Good afternoon, everyone. Welcome to the 1 o'clock session, Making a Case for Host Response Diagnostics, Real-World Impact, Workflow, Integration, and Economics of LIAISON MeMed BV. My name is David Engman. I was professor of pathology at Northwestern Medical School for 25 years and chair of pathology at Cedars-Sinai Medical Center in Los Angeles. This showcase is sponsored by DiaSorin, and the speakers today are Brian Duchateau and Amin Mohammed. Brian received a PhD in bacteriology at the University of Wisconsin as a board-certified medical laboratory immunologist. He began his career in clinical diagnostic laboratories, but in 2009 moved to the IVD industry and worked for a number of companies before joining MeMed recently as U.S. Head of Clinical Affairs. Dr. Mohammed rose up the academic ranks at UTMB Galveston, West Virginia University, and Texas A&M Health Science Center, and since 2010 has been in Temple, Texas. He's a clinical chemist at Scott & White and is chief clinical chemist and director of pathology informatics. Now, this is a two-part presentation followed by a panel discussion, and we'll begin with Dr. Duchateau discussing the MeMed BV test and everything you need to know as background before Dr. Mohammed's presentation. All right, Brian?
So welcome to ASM. It's one of my favorite meetings of the year. I'm glad to participate this year and talk to you about a new technology.
Can I borrow the pointer? Thank you.
A new technology that we call the MeMed test. And I often get asked, what is this new test with the funny name? And really at its very essence, what it is a test that helps practitioners tell if their patients have a bacterial or a viral infection. Now, of course, that's a very simple question to ask, but often clinically a difficult one to answer. And of course, there's a lot of chemistry and immunology behind this test, but at its very essence, that's exactly what this test does. This test was FDA cleared in 2021. And our marketing people say that it was a landmark clearance, and maybe that's a little bit sensationalized, but it is unique in that it was the first test cleared by the FDA in a whole new category called host immune response testing, and MeMed helped the FDA really define that category. So when we're diagnosing a patient right now with, whether they have a bacterial or viral infection, important question to answer, one you can readily treat, one you can't so readily treat. The way we do it today is we utilize all of these kinds of tests. And I know that many of you that attend ASM are laboratorians, and so a lot of these tests will be familiar to you. There's nothing wrong with these tests. These host immune response tests are not going to replace these types of tests, but they will be adjunctive to them. And the reason that we need adjunctive tests is because these tests are associated with limitations. We'll talk about those in the next slide. But I mention all of this because I really think that host immune response testing, this might be the first time you've heard about it, but it won't be the last. This is not, in my view, a technology that's flash in the pan, here today, gone tomorrow. I really think host immune response testing is here to stay. And it's here to stay because of all of the limitations associated with these conventional tests that we know. I won't say love, but we know. And so that's what's really driving this whole development of host immune response tests. Here's some of the limitations, and I won't go through them all because I know most of you will know and appreciate them. One of the limitations is prolonged time to results, especially if you're talking about culture. Think how long it takes to get a conclusive negative blood culture back, right? Long after the patient has been triaged through the ED or the urgent care center. One of the other big issues with those conventional tests is right here in the middle. About 50% of all community-acquired pneumonias never even have a pathogen identified. We just empirically treat them. One of the things that's very popular right now is to utilize these molecular viral panels, the most common one being what we call the quad panel, SARS and flu A, flu B, and RSV, the 4. 42% of those are negative. That's a diagnostic dilemma, right? Because you still have a patient that's sick in front of you. You know that the 4 things you tested them for are negative, but you don't know what they have yet. You still don't know whether you should treat them or not. A lot of those conventional technologies also don't do a great job with bacterial viral co-infections. This was a particular problem last year's flu season. Depending which geographic area you were in, about 20%, up to 20% of all flu A positive cases had a concomitant bacterial pneumonia, underlying pneumonia. And so that's also a diagnostic dilemma, because you run a PCR test, which is exquisitely sensitive, exquisitely accurate. You get a flu A result, and you think that's your answer, not knowing that you've missed a bacterial pneumonia, and that patient ends up coming back 3 or 4 days later, much sicker than when you first saw them. And one of the other problems with common pathogen detection like PCR is just because you detect a pathogen doesn't mean that's the etiology of disease. And that's a problem in carrier states, especially in children, like with group A strep, with adenovirus, things like that. So these are all problems that I won't say host immune response testing solves, but it certainly goes a long way to addressing these gaps. That's why I think this type of testing is here to stay. I don't often put this slide in my presentation, but I thought I would do it today because I know a lot of you are laboratorians and have a great grasp on technology. This is how this test was developed. So MeMed started with over 100,000 different protein candidates. Just think about that for a minute. It's only about 20,000 genes in the human genome. But if you look at all of the different isoforms that are available, there's about 100,000 different proteins. And so MeMed started with almost a comprehensive list of almost every host biomarker or host protein, and then used machine and AI learning to pare down the number of proteins, and then took on a Herculean task of developing 600 different ELISAs, actual tests in the laboratory, and started to develop heat maps of which biomarkers were expressed in individuals with characterized bacterial or viral infections. And kept doing that, whittling down the number of proteins, increasing the number of patients, whittling down the number of proteins, increasing the number of patients, until they got all the way down to these 3. And we looked at whether the test should include 17 proteins, should include 30 proteins, should include 5 proteins. And no group of proteins ever outperformed these 3 host biomarkers. And so these 3 became what we call the MeMed signature. The way the test works is the assay simultaneously quantifies all 3 of these markers down to the picogram level, so exquisitely sensitive, and then it puts the quantitation of each one of these markers into a proprietary algorithm, and the algorithm spits out an objective score of between 0 and 100. And that score correlates with the statistical likelihood of the presence of a bacterial infection. So really high scores correlate statistically with a really high likelihood of a bacterial infection, and really low scores correlate with this really low statistical likelihood of the presence of a bacterial infection. And although this assay has really good positive predictive value, in the mid-90s, its strength is its negative predictive value. It's like 99.3 and change, which means if you have a very low value, you can hang your hat, or almost hang your hat, that there's no bacterial infection present. That's the true power of the assay. The assay is FDA cleared on several different platforms, 2 DiaSorin LIAISON platforms, and also a MeMed key instrument. The DiaSorin platforms are your conventional lab analyzer, high throughput. The key instrument is more of a near patient solution. Whole blood and serum are both cleared on the key, and serum is cleared on the LIAISON. This is a slide that represents the clinical trial-like studies that have been done on the MeMed assay. And I won't go through all these obviously, but a lot of IVD companies don't even publish one clinical trial. Here's 8. Apollo was the last one, which was the data that was submitted to FDA. And what I think is most impressive here is not that there's 8 trials represented here that are published in really good journals, but what I think is really interesting is that these assays or these trials are almost 10 years apart. Curiosity was about 10 years ago. Apollo was just published last year. And even though there are a decade of time represented here, different studies in different cohorts with slightly different protocols, the performance of the assay has remained consistent over time and different patients. To me, that's what's most important. With that said, MeMed has really gotten away from doing these types of studies. These are the types of studies that show that your test does what we advertise it to do. It does what it's purported to do. And we've gotten away from that because what people really want to know is at some point they start to believe, yep, your test does what the package insert says it does. But really what we want to know is if we utilize it in our institution, how will it benefit the institution? How will it benefit the physician that utilizes the test? And most importantly, how does it benefit the patient? That's really what they want to know. And so we've spent the last 18 months or so generating real-world evidence, clinical utility, and clinical outcomes data to answer those types of questions. By any stretch of the imagination, this is the most well-studied host immune response test on the market. There is almost 60 peer-reviewed publications now, over 200 clients, and several 100,000, this is an outdated slide, of tests that have been run. MeMed is in a really good spot because the types of clinical utility and clinical outcomes data that I described in the previous slide are not even being generated by us anymore. They're being generated by our customers, which you're going to hear from in a minute. And some of the things that they're showing is that the assay can modulate length of stay in the hospital, that there's a cost savings per patient when physicians ally their practice in accordance with the MeMed test result. There's fewer admissions. We even have customers that are utilizing the test to better optimize which patients they do that expensive multi-panel PCR testing on. And we're seeing a reduction in the use of antibiotics, especially in patients with viral infections, which is something that we should all be cognizant of. 2 slides left. And I could have selected any one of these things to tell you about, but I only had a few minutes of time allotted. So what I chose to do is give you some of the newest data that's coming out. This is from Tampa General. It was just presented at the SAEM meeting in May. And what they did is they incorporated the MeMed test directly into their sepsis order panel. And they did it in 2 phases. One was operational, where people in the sepsis panel were getting the test, and the result existed, but the doctors didn't get to see it. This is essentially the control group. And then interventionally, where the doctors were actually trained on the test, what the test result meant, and got to react to the test result. And if you're familiar with the new Surviving Sepsis campaign, they want us to categorize sepsis patients into 3 different categories, actually 4: definitive and probable, possible, or unlikely. And if you look at the MeMed test results here, it didn't have any statistical difference in individuals with definitive or probable sepsis. That's good. And the reason it's good is because we don't really want the test to be utilized on people that are toxic looking, that have definitive sepsis, that are hemodynamically unstable. You don't have time to run a MeMed or any other test. You've got to save that person's life. What we really think the test is most valuable in is where there's diagnostic uncertainty, is where you're not sure what the patient has. And here you can see that the test did effectuate some change here by decreasing the number of patients with possible sepsis and putting them in the unlikely sepsis category. That's important because as you reduce unlikely sepsis, You get these people out of the SEP1 protocol, which lowers the denominator and hopefully makes people's SEP1 metrics look better. Of course, when you do this sort of thing, there's a financial implication for this. This is Tampa General's financial implication. You know, less admissions equals less bed days. You multiply that by your standard day rate, and there's a cost savings of about $1,700 per patient. $1,700 per patient times thousands of admissions in a year is big savings. And this isn't the only institution that's showing this. This is triangulated in many different studies. You're going to hear about one right now. I think we should save questions till the end.
All right.
All right. I would like to start my talk by first thanking to James and Ileana for giving me an opportunity to talk about our experience with the MeMed test. The name is really funny because when I went to my LIS guys and told me that I want to build a MeMed test, they said, "What the heck is that?" How do doctors will know that this is a test to order? So we changed the name to bacterial viral protein profile. So if you come to the Baylor Scott and White, your physician there, that is what you will see in our EPIC chart. Okay, so we have been looking at the MeMed for quite a while, and our journey started maybe about 3 years ago, 2 to 3 years ago. And we have been looking at the literatures, we have been talking about it, we have been discussing about it, and looked at so many publications that they have published on this topic. And I personally went and discussed with a lot of our emergency room docs, with our family practice physicians. Everybody has some sort of a use case. But one of the most important use cases that really stood out for us at Baylor Scott and White was inpatient. Now this may sound really very weird for you guys because its obvious rule is to rule out a sepsis or infection in the ED room. You'll save a lot of money. Other obvious use is to go to the clinics and then to find out whether the bacterial or viral, and you can cut down on using the antibiotics, give the patient that. But when I was talking to my hospitalists, they said that, we have a problem in our hospitals where we need to turn the beds over quickly. And Baylor Scott and White is about a 50 hospital system, and where I work in Temple is one of the flagship hospitals. So we always have the problem about how to discharge the patient quick enough. And one of the area of that problem is, when a physician suspects an infection, they have to get the cultures done on the patient, and they have to wait for 5 days to get the culture results. Sometimes they start antibiotics, and they are hesitant to stop the antibiotics at that time. So the beds are not turned over quickly. So these are some of the diagnostic. I'm sure that you all have seen this. I'm not going to go over this in detail, but you all are laboratorians, all work for the hospitals. You are aware of the challenges that we have in our hospitals. So the main thing that, how does the MeMed test, when I started talking to the hospitalists, they were actually very much interested to find out that if we can know what is the viral infection is in a patient, then we can de-escalate the antibiotics, or we can not even start giving them antibiotics. Our discharge protocol is a lot easier and simpler as compared to the protocol when you have a bacterial infection or suspected bacterial infection protocol for that. So with that, we started this journey. We started talking with the MeMed, trying to acquire the instrumentation and the test, and also to get the funding for that part of it. So luckily, we were able to get the LIAISON Excel machine for that, and we went through the MeMed validation process, which was really quick, and we got a help from the MeMeds at that stage. I asked for him to get the samples, do it in our lab. The EPIC build was one of the most time-consuming part of it, just to get them to build the test into the EPIC and to make it work, get all testing done. That took us about 6 months or so. Then we selected a go-live date and passed out a laboratory announcement. And the most important thing that we did was we started having a multiple lunch and learn with our hospitalists. And we took upon us that I took the responsibility of going and talking to the physicians. Of course, LIAISON helped us out with getting some lunches and all, gave a lot of seminars within our system. We not only included the hospitalists, we also included physician assistant, nurse practitioners, infectious disease guys, and we keep on doing this from hospitalists to ICUs to emergency room medicine. So that was extremely important. And like any other test, when you bring it in, the volume increases slowly. You know, in the beginning, the physicians were not too certain about it. But then as time went on, it kept on going up. And then all of a sudden, you know, what happened, what I believe what happened, is the physicians who started out using this test saw the results for it. It helped their patient. And they started talking that anecdotal instances to other physicians. For example, one of the physicians was saying that, you know, he had a patient that he saw in a lab in his hospital, that he was thinking that the patient had a viral infection. And, but before he thought that he's going to discharge the patient, let's just do a MeMed test. That turned out to be finding out to be that it was high bacterial. So he did, so he did an imaging analysis and he found that the patient had pneumonia. And the patient was started on antibiotics and patient recovered. So those type of stories started being exchanged between our hospitalists. And then all of a sudden, I was amazed to see that the volume just goes up. What was important that for me was, as you can see, that they're beginning to add ordered MeMed tests independent of the culture. That, you know, of course, they're ordering culture and the MeMed together, but this number of volumes of the MeMed, just MeMed by itself is increasing. So after about 5 to 6 months, you know, whenever we bring up new tests in my hospital, it becomes the responsibility of me as a laboratory director to see that how the test is performing, whether it is coming up, meeting the goals that we wanted to meet for our institution. So we went into we are lucky that we have this population analyzer software, which is connected to our Epic. And what we do in that analyzer, in that particular software, it is basically a database. And you can go into that database and you can extract all the information you need. So I was able to download all the MeMed testing that has been ordered by the physicians. Also was able to download all the cultures that were ordered during the same amount of time, and then how many samples had both the cultures as well as the MeMed was done. The reason I did that, I will tell you, as you see the data, it will become apparent why I went through all of that workload. So first thing, so we divided them into 3 groups. Group A was just the cultures only, and Group B was culture plus MeMed, and Group C was just the MeMed test. And if you look at a Group A, there were 8 patients were positive blood culture, 154 were negative. And what I was interested in to see the length of stay, because we were not worried about the performance of the MeMed test, its sensitivity, specificity, negative predictive value. It has been published ad nauseam. Very good journals, very good institutions. So I was not going to worry about challenging the test accuracy. What I was worried about, if I bring this test, will it be abused by our physicians, or is it going to be able to provide the support that we needed for that, or the outcomes that we were looking for. So in a Group A, the length of stay was about 11 days average. This is a confidence interval for that. Negative patient was 7.8. That is to be expected. I'm not going to go over this because Brian did a wonderful job in talking about how the test is being utilized, the scores. The next thing I wanted to find out when I was looking at the data, is it that I have somehow the patients, they've been distributed equally in terms of their mortality morbidity score, how severe the patients are. So we have in our hospital we use this Elix-Hauser comorbidity count and Elix-Hauser mortality count. What does that tell you? How sick the patient is, how many comorbidities they have. So we took all the 3 groups of patients, we downloaded that information, and we did a statistical analysis. What we found out, there was no statistical difference between the 3 groups. The only thing that we found out, which was of maybe for the future studies and future work, is that we found that Elix-Hauser score for the equivocal patients was relatively higher than the other one. Most of the equivocal patients had the higher Elix-Hauser. Thank you so much. So most of the equivocal patients had a slightly higher, but that is something that we are going to investigate later on to see those part of it. What is with that? So these were the key findings that we had from our database. One was there were 13 patients total that were positive by culture. Out of that, 11 patients were also positive by MeMed test. And the 2 patients had an equivocal result, that MeMed was not able to say whether it is bacterial or the culture. So the accuracy of the test turned out to be 100%. When culture is positive, the MeMed was positive, which is very important for our physicians to know that. There were about 58% of the patients that are negative bacterial culture, but were the MeMed positive. Now that is important because that there is some sort of infection is going on that we are not catching by the blood culture. Third one was 27.23% of the patients were viral positive, or they either were viral positive or they were negative. We are not sure 100% for that because we didn't do the viral RVP panel on that. And then there was 15% that had a negative blood culture, that had very equivocal results. So now we started comparing the length of stay for the people that they had a MeMed test was done on it. And what we found out, the patients that had high bacterial probability, the length of stay was about 7.9 days, interval 6.76 to 9.04. High moderate viral length of stay was 5.19. Interval was 4.38 to 6. Equivocal was about the same as that of the high bacterial count. What that showed us, that the length of stay difference between the bacterial and the viral likelihood MeMed result was statistically very, very significant with a p-value of 0.02. Also shows us that on an average, we were saving about 2 to 3 days reduction in the length of stay. Now that's a very interesting finding that we were able to find in our hospital. And this is like, we just took the test, we taught the physician how to use it, and we told them, if you suspect infection, order this test. And then we sat back for 6 months. We didn't coach any further coaching or training, and we just looked at the data. And that is what is based on the data that we are finding this. What were the clinical implications for that? The clinical infection is that, about 57 point with a negative bacterial results, culture, which shows that, about the MeMed was positive, there's some other source of infection going on in the bloodstream. Early antibiotic treatment before the culture collection. That could be the reason for that, and the false negative blood cultures. In our studies, we found that the blood cultures were positive about 6 to 7% for all the blood cultures that were collected. Patients with a viral likelihood, MeMed results tend to have a shorter hospital stay, about 5.2 days. Patient with a bacterial likelihood at 7.9. Then we also had in this database access to the direct cost. So how much it costs for my organization to take care of the patient. It is not the charges, it is not the amount of money we got back. how much cost we incurred in taking care of a patient. And what we found out, and I started looking at that, and the direct cost for, the direct on an average for the bacterial infection was 12,000, 6,000, around 6,000 and 14,000. Again, the comparison between the statistical comparison was very significant. The p-value is 0.004. So from this, we were able to show that there's about close to about $6,000 savings, because without the MeMed test, what was the alternative? You wait for the culture, wait for the results to come from the culture, wait for the continue doing the antibiotic treatment. So that option was not even there. So that's the reason why we feel that savings. Other thing was, we also, now we my attention was turned to the group C patients. So what is happening with them, where they didn't order any culture, but they just ordered a MeMed test. How did that work out? So again, on the top is the average length of stay, and at the bottom, it is the median length of stay. And the reason I did that was, average can be skewed, as you know, by the outliers, where the median is telling you what is the halfway point. So what we are finding out again, for just doing the MeMed, the length of average was about 6.25, 4.1. It's the same pattern, about 2 days savings in the length of stay was repeating itself. You see the same thing for the median days, it was the same. So here is a side-by-side comparison of this. Now the third point, which was, I got a little bit excited about this, and I showed this data to my department chair and to my chief financial officer for my hospital, because it was important. You bring in a test, you spend money, you do the validation, you work through all the LIS. We want to make sure that the test is having some impact on your healthcare system and the hospital. So I was able to share that, and one of the questions that were asked to me at that time was, has it impacted antibiotics use? So we looked into the group B patients where actually the antibiotics has been actually, where the MeMed and culture was done at the same time, and the group C where there was only MeMed testing. What we found out in that case was, in a group B, there was about 10% reductions. There were a total of 6 patients in the group B with just the culture and the MeMed, where the antibiotic was stopped for the 2 patients, and the 4 patients that were not given antibiotics. Now here you may be asking that what time was this test was ordered. The culture and the MeMed was ordered within about 1 day of that. So it's on the same day those tests were ordered. In the group C, as soon as they got the results, and next question was obvious, what time they stopped, if they stopped giving the antibiotics, what time they made the decision not to give the antibiotics. Again, it was within 1 day. They're very close to each other. There was 76%. More importantly, there were 28 patients that were not given antibiotics, and there was 4 patients, the antibiotics were stopped. Now, I didn't really, if you look at the direct cost, I didn't separate it out what was the antibiotic cost. I know some of you guys are pharmacists over here. Some of the antibiotics that we use for the inpatients are pretty expensive. And so I didn't separate that out in my database, but it was lumped into the total savings. So the likely driver for the reduced length of stay and the lower cost, that was an obvious question. And early antibiotic discontinuation or avoidance, that's one of the likely costs for reduction in the direct cost. Less diagnostic uncertainty at discharge. Now the physicians are more confident that, if they suspect the patient to be a viral patient, the patient is getting better. And in the old days, they used to wait to get to get the culture results back, which may take up to 5 days. So that was a reduction in 2 days. That's where it helps. It's lower need for resource-intensive bacterial infection management. It's relatively lot easier to take care of the viral infected patients versus the bacterial. There were fewer discharge barriers. Remember, I was saying that a hospitalist, one of the questions with the hospitalist is, when I was talking about this test and they came back was saying that, this test may help us in getting the discharge done faster in the hospital. And then the most important, that the Elixhauser score that we are looking at, it is just like, the shorter length of stay and the cost may be related to the infection type, classification, and care pathway. What we are observing, there is based on the MeMed results, the physicians are developing their own care pathway for treating those patients. Now, this is not something that we taught them. This is not something that we told them that this is what you should do, this is what you do. We just provided them the test. We told them to use it for management of infection patients, and they are themselves developing these pathways which are showing a significant decrease in the length of stay and the direct cost saving. So finally, I'm showing you the data just for the inpatient. So we are showing that, when the hospitalist-based hypothesis was, it did decrease the length of stay for the people who are positive for the viral versus that of the bacterial. And then fewer unresolved culture negative infection questions at the discharge. That constant nagging thing that a physician has in his brain that, whether it is a bacterial or a viral, this test was able to help them resolving that question. That was very important for our hospital. So where are we going to go with this? Where are we going to go with this, with the next thing? So this is like a continuum. In the hospital of my size, which is a tertiary academic medical center, we have to educate the physicians, which we are doing. So we moved from the hospitalists, we are now getting the pulmonologists, the ICUs. We are seeing an uptake of MeMed tests. Big uptake is occurring also at the ED room. Maybe in about next 6 months, data will increase a lot of patients from the emergency department. Our physicians, one thing that I found out I've been in this business for about 34 years as a lab director. The one thing that I feel the physicians are the community which are the least resistant to take new technology. And one of the ways that at my hospital that we are doing it, when we get a new test, we actually bring the test in-house. We let the physician use it. We tell them what the publications are telling about it, and then they use it in the appropriate situations. And we have found that strategy to work very well. And as we have seen in the case of the inpatient. And so we are hoping that not only my physicians at the temple, but even the physicians at our system level at other hospitals are going to start using this test.
Very good.
Any questions?
Thank you. So we have about 10 minutes for a panel discussion, but I think it's very important that the audience questions be answered at any
We have a
time.
microphone, too.
And there's a microphone, so raise your hand and they'll come around with the microphone. Let's go up here in front.
So thank you so much. That was great in terms of both the technology and the clinical side. I'm curious that one of the problems that you brought up at the beginning was the challenge of co-infection. Is there any way to say give 2 scores? Because one could interpret the middle ground in lots of ways. It could be a co-infection, it could be no infection, it could be a technical error. Is there any way that you can actually give 2 scores in the future with this to have an independent look at viral probability and bacterial probability?
Dr. Duchateau?
Is this on? Yep. I think it's better here.
He'll turn you up. He'll turn your mic up if you sit here.
Okay.
Yeah.
Can you hear me okay?
Hear me okay? So the test is actually engineered so that if there's a co-infection, it defaults to a bacterial result. So the bacterial result will trump the viral result. That was done purposefully because we thought that the bacterial result is more clinically important and you have more intervening therapeutic options than you do with the viral result. I'll add one little anecdote that we didn't go to the FDA thinking that we were going to be able to get a co-infection claim, but we happened to have so many co-infections in our trial, and the data was so good that the FDA allowed us to make a co-infection claim.
Very good. So let me ask a question then. Are there others here? There's one here. Thank you.
Very nice presentation. So I have two questions for the first speaker. So you mentioned that this test is showing a higher level of 3 markers in bacterial infection and lower level in viral infection. Is that applicable to all type of patients, like including immune-compromised patients, like transplant, patient on chemotherapy, diabetic, and so forth, or just like immune-competent individual?
This is a great question. And if you've read our package insert, one of the limitations that is listed is people that are immunosuppressed. And it's listed as a limitation, not a contraindication. And it's listed as a limitation because you can imagine there being a person that's immunocompromised to the degree where they have an inability to mount a host immune response. And if they have that, then that would effectuate or modulate the MeMed test result. The reason, but also if you think about an immunocompromised patient, they're not all created equal, right? So, you know, somebody that has chronic variable immune deficiency or SCID is much more immunocompromised than somebody that took 2 puffs of a corticosteroid, right? And so it would be impossible for MeMed to test all different types of immunocompromised patients. But you can imagine there'd be a point where they could be immunocompromised to the point that it would affect the assay. With that said, what we've done is we've gone back and we're running studies right now and clinical trials on what we think are the most common types of immunocompromised individuals that are encountered. So people with solid tumor malignancies, people on chemotherapy, transplant patients, solid transplantations. And has been no difference so far. You're going to see some of those publications come out from Johns Hopkins. Tampa General just reported data at the SAEM meeting in Atlanta 2 weeks ago. So you're going to see those types of data. But so far, so good on the most commonly encountered types of immunosuppressed patients.
All right, so let me ask a question now. Dr. Mohammed, you know, most of the inpatients in your study were tested within a day or 2 of admission, but what other inpatients might also benefit from this test?
What other inpatient might have benefited from this test?
Could benefit in the future.
Could benefit from this test. Again, you know, from the inpatients who are from the surgeries, they have just had an operation surgery and they are being taken care by our hospitalists, and all of a sudden they start developing fever, and that is also a suspect at that time whether it is a bacterial fever or the hospital-acquired infection. This test can be very good in terms of doing that, finding out is a bacterial infection, especially when we know the accuracy of the bacterial infection for this test. So those are the one group. Patients we are seeing in the areas like orthopedic areas. There also when they have got the infection in the bones, or when they have any kind of the fractures and all that, they are being treated. Other area is also the pulmonology, where the physician is seen, or where patient is seen in the pulmonology, and they do an X-ray and they find the fluids in the lung. And now they have got a suspect whether this is due to the congestive heart failure, for which they can do the BNP, NT-BNP, or they want to find out it is not due to the infection. So there is another area where they can do the testing for this one. Of course, ED room is the next big one where we are doing the training for our physicians to order this test. Hopefully, we are able to put it as a sepsis order set, part of the sepsis order set for doing this. So right at the moment the patient walks into the ED room, they suspect the patient may be a sepsis patient, run the test along with all the other ones they're doing. Like, they do the lactic acid and the CMPs and the CBCs.
Dr. Duchateau, there's a question down here. Yeah.
Yeah, so I imagine a lot of the data behind this test is for acutely ill patients presenting to EDs. Do we know how it performs in the markers and the test itself performs for chronically ill patients or those with an indolent bacterial infection?
Yeah. The short answer is, if you look at the package insert, it says that you should utilize the test wherever there's a suspicion of an acute infection. And so all of these kind of chronic issues, I'm not sure how the test performs. I'm an immunologist by training, so you can imagine that when you have a chronic condition, your immune system kind of just settles down and dissipates, right? And so I'm not sure how the test would perform in those chronic conditions.
All right. Dr. Duchateau, I have another question for you. So you're familiar with this test at many hospitals and clinics since you're an advisor to MeMed.
Yeah.
Where is the test used most, and what do you see as the future sort of expansion of the types of patients who might benefit from this test?
Yeah, it's another great question. And this, we're starting to get, I mentioned in one of my slides that we have over 200 customers now, and if you'd asked this question to me, 2 years ago, I'm not sure I would have been able to answer it. But the way it's working now is about 35 to 40% of our customers are urgent care centers. They, just like ED, they have a need to triage patients quickly, get them through and identify who needs to be treated and not. About 60 to 70% of our customers or so are hospitals. It's being utilized most right now in the EDs. And about half the people in the hospital accounts that are using it are ED physicians, and about half are hospitalists.
Very good. My final question, unless there's one from the audience, is for Dr. Mohammed, and that is for other hospitals considering implementing this test, what did you find were the greatest barriers and what helped you to overcome the barriers? Whenever we all bring in a new test, we all face this, but what's your advice for new hospitals?
Yeah, in our Baylor-Scott system, we have 51 hospitals. They are all of different sizes. There are some which is 50 beds, some which is 120 beds. There are some which is 300-bed hospital. So one of the main things, our main finding after this test is that the reduction in the length of stay. Nowadays, I didn't stress upon this a lot, but to get the patient moving in the hospital is one of the biggest challenge to decrease the length of stay and to use the hospital capacity to the maximum extent. The data that we have got in this particular slide, that in this presentation that we found, that 2 days decrease in the length of stay is a huge deal. So as soon as I passed this data on to my department chair, and then it was given out, distributed to all the laboratory directors within my system, I've been getting so many calls to give me the contacts, asking questions about exactly what Dr. Ingman is asking, the laboratorians. One of the big thing about the laboratorians that for us is if we bring in a test, we want to make sure that it is have a positive impact on our patients, on our hospital system, both economically and also we are making a difference. So the barriers that we have got nowadays is obviously the space. Everybody cannot have a lab space to have an EXL, which is a relatively bigger chemistry kind of analyzer. The second thing is a cost, but the space issue is solved by having the key instrument that they have got, which is a smaller machine that can be put. Yeah, it is problematic that there's only one test at a time. You cannot have multiple tests. Cost issue, I hope that our experience with the cost savings of $6,000 on each patient, that's a huge amount of savings. So return on investment cannot be very easily calculated, and it can be monitored. The most importantly, you can actually develop a metric and that you can compare that and see whether the test is performing that level or not. And that is the most important thing. So when you go to the CFO, when you go to the CEO or a hospital administrator or director, you can tell them, my guys are able to go and talk to them and say that this is analysis, this is how much of savings we are projecting by doing this testing. And that barrier comes down at that stage. The third barrier, which I can tell you, is going to be your LIS system. Working with the LIS system is going to be the most critical part. That's the most rate limiting area, because we always have infectious disease patient in the lab, in the hospital. Getting sample is not a problem. Getting an instrument from these guys, from the DiaSorin guys, getting the kits is not a problem. But getting it implemented, building all those LIS barriers, that was my biggest barrier in my lab.
With that, we thank the audience and thank our speakers for the session. Thanks.